Welcome to week 9. So, in today's lecture we are going to talk about isolation and characterization of DNA. In a previous lecture, that is in week 7, we talked about isolation and characterization of proteins, and today we are going to look at isolation and characterization of another biological macromolecule, which is DNA. So, in today's class we are going to talk about DNA, which is the genetic material, and how to separate that from other cellular components just like protein, and we will see that this isolation depends on the type of DNA and also from the source and size of the DNA. So, before we go into the actual experimental details, let me step back and talk about the basic central dogma of molecular biology. So, here what we see is the central dogma of molecular biology, which was first proposed by Sir Francis Crick, who along with James Watson proposed the double helical structure of DNA. So, what is the central dogma of molecular biology? It tells us the flow of information, that DNA is the genetic material which stores all the information that is necessary to carry out all the functions in a living system, that is a cell or an organism. So, DNA codes the information, and on the other extreme we have protein which carry out all the function. So, proteins, like in the previous lecture we talked about enzymes. Enzymes are catalysts, and then there are also other types of proteins which are not enzymes, but then they also carry out different functions like receptors on cell surfaces, or there are cytoskeleton proteins. So, there are so many different types of proteins which carry out most of the functions inside a cell. So, this is the functional part or the functional macromolecule, and this is the macromolecule which stores the information. In lecture 7, we saw how we can isolate proteins and do characterization and characterize them using various biophysical methods. Today, we are going to look at DNA. So, DNA stores the information. When a cell divides, the DNA also has to multiply. So, that copying of DNA is called replication, and it is done by enzymes which are again proteins. These enzymes are called DNA polymerase. Now, once you have the DNA, if you want to make the protein, you have to extract out the information. So, when we talk about DNA, we are talking about the full DNA. For example, in case of bacteria, let us say E. coli, the DNA is several million base pairs long. So, typically it is around 5 million base pairs long, but that long DNA has information for different types of proteins. So, if we want to make one particular protein, that information might be encoded in only 1000 base pair long stretch of DNA. So, that particular part, which is referred to as a gene, is copied into a temporary copy called the RNA. So, both DNA and RNA are nucleotides. So, we will see in more details what we mean by nucleotides. So, a small part of DNA is copied into RNA, and this process is called transcription, and it is carried out by another type of enzyme called the RNA polymerase. So, this small part of nucleotide, this small nucleotide has the same information as that of the DNA, but this is only a small part. So, this is only 1000 base pair long, which encodes information for only one protein. Then that information is read by another machinery called the ribosome to synthesize proteins, and this process is called translation. We have seen that once the protein is formed, it is a linear chain of amino acids. So, the protein is a polymer of amino acids, and then this polymer spontaneously folds. Similarly, DNA and RNA are also linear polymers, and they are polymers of nucleotides. So, there is a different type of monomer, different from the amino acids. Again, there are 20 different amino acids in case of proteins, but there are only 4 different types of nucleotides in case of DNA and RNA. One major difference between DNA, RNA and protein is in the shape of these molecules. So, we have seen that once the linear protein molecule is synthesized, it spontaneously folds into a 3-dimensional structure. So, we looked at that in the lecture where we studied protein folding and unfolding. DNA on the other hand has a very unique structure. So, it does not depend too much on the sequence of the DNA. All the DNA molecules fold up into a structure which is called the double helical structure. So, typically most DNA have this double helix structure. If you see carefully, you will see that there are two polymer chains which are intertwined with each other. So, that is why it is a double helix. RNA is a single chain. So, that is the difference between DNA and RNA, and we will see some more differences, and we are going to exploit those differences to purify DNA from RNA and proteins, because in a cell all of these macromolecules are present at any given point of time. So, what are nucleic acids? This is the typical structure of a nucleic acid. So, these nucleic acids are polymers, and this is the monomer of a nucleic acid, and these monomers are called nucleotides. If you see, there are three different components in this nucleotide. The first component is the nitrogenous base. So, this is the nitrogenous base, and there are two types of nitrogenous base, purines and pyrimidines, and again we will see in the next slide there are two purines and two pyrimidines. So, there are four different types of nitrogenous base which is shown up here. Then the second component is the sugar moiety. This ring like structure is the sugar moiety. Depending on whether this is a DNA or a RNA, the sugar is deoxyribose or ribose. The difference between these two sugar is in this carbon. So, this sugar molecule has 5 carbons. This is 1, this is 2, this is 3, this is 4, and this is 5. At the second position, you will see that both are hydrogen atoms. So, this is deoxyribose. In case of ribose, this H which is at the bottom, this H is OH. So, that is the ribose sugar, and when this oxygen is removed, it becomes deoxyribose. The third component is the phosphate group, which is attached to the fifth carbon of the ribose or deoxyribose sugar. These two, when you have only the nitrogenous base and the sugar, so the nitrogenous base and the sugar, if you have only these two and not the phosphate, then it is referred to as a nucleoside. When you also have the phosphate present at the 5 prime position, then this monomer is referred to as a nucleotide. So, in today's lecture we are only going to refer to nucleotides. So, now coming to all the 4 different nucleotides, that is the monomers of DNA. So, the one that we saw in the previous slide was this, which is known as deoxyadenylate, or in short A. So, we just like the proteins where we had 20 different monomers, and each of the monomers were given 20 different letters from the English alphabet. Similarly, in case of DNA, the 4 monomers are giving 4 single letter codes. The first one is deoxyadenylate, and it is referred to by A. The second one is deoxyguanylate. You see that the base is the one that is changing, and it is referred to by the letter G. Third one is deoxythymidylate or T, and the fourth one is deoxycytidylate or C. If you see carefully, the sugar and the phosphate groups are exactly the same in all these 4 nucleotides. It is only the base which is attached at the first position of the sugar, this base is different. So, it is only the nitrogenous base that is different, and between all these 4 monomers. Now, these monomers are attached just like the proteins, where the amino acid monomers are attached to form a polymer. In case of DNA and RNA, these nucleotide monomers are attached to form a linear polymer. So, on the left hand side, you see this linear polymer is that of DNA, and on the right hand side, this linear polymer is that of RNA. So, if you compare them, you will see that the difference between these two is in the nitrogenous base. So, here the second position has H, and here the second position has OH in case of RNA. So, this is the ribose sugar and this is the deoxyribose sugar. Another difference that you will see is in the bases. We will come to that later. So, how does this polymer form? So, this is the monomer, the base, nitrogenous base attached to the first position, the ribose, the deoxyribose sugar, and then the phosphate group attached to the fifth position. Now, you see that another phosphate group is attached to the third position. So, the third OH, and this is connected to the fifth CH2OH of the next nucleotide. So, this forms the phosphodiester linkage between two monomers. Similarly, there is another phosphodiester linkage between these two monomers, and it proceeds so on and so forth. This phosphate is attached to the 5 prime carbon. So, this is referred to as the 5 prime end, and this phosphate or this OH. So, this is the 3 prime carbon and this is referred to as the 3 prime end. So, just like a protein where we have the N terminus and the C terminus, in case of DNA or RNA we have the 5 prime end or the 3 prime end. So, I was referring to another point that apart from this 2 prime position, the nitrogenous bases are also different. So, A, G and C are exactly same in case of both DNA and RNA, but only the T is something that is different. So, instead of thymidine we have uracil in case of RNA. So, it is one of the bases that is different, and this 2 prime position where it has a OH in case of RNA and H in case of DNA. Another difference between DNA and RNA is that DNA forms a double stranded structure. So, it is shown here, this is one of the strands. So, you see it runs from 5 prime to 3 prime direction, and this is another strand which runs in the opposite direction. So, this is 3 prime and 5 prime. And what happens is the base C and base G, they can form hydrogen bond. Similarly, base A and base T, they can form hydrogen bonds. So, these hydrogen bonds, this pattern holds these two strands together, and we have a double stranded DNA, and we will see in the next few slides that these strands forms a spiral like structure which is referred to as the double helical structure. This type of double helical structure is absent in RNA, because RNA mostly they are single stranded. This is something that is very important, especially for today's lecture, that RNA, it has this OH at the 2 prime end, and the presence of this OH makes it chemically less stable compared to DNA, because the presence of this OH results in a particular reaction where a hydroxyl ion, which can be supplied by high pH or it can be also enzyme catalyzed, this hydroxyl ion can attack this, so that there is a nucleophilic attack and this phosphodiester linkage is broken. So, that now you have a breakage in the RNA strand. So, this type of reaction cannot happen here because we do not have an OH group here. So, this makes DNA molecules much more stable chemically compared to RNA. Moreover, the double stranded nature of DNA makes it even more stable compared to RNA. So, we are going to exploit this particular difference in the chemical nature of DNA and RNA when we are going to separate the DNA and RNA molecules. So, as I mentioned before, the two strands of DNA can form a double helical structure like this. So, you can see that one strand is this. So, you see it is a helical structure. So, it spirals, and the other one follows it and forms another spiral. So, together they form a double helix, and the nitrogenous bases are all inside, whereas the sugar and phosphate which forms the backbone are on the outside. So, this results in a highly negative charge on the DNA molecule, because all the phosphate groups carry one negative charge. So, deoxyribonucleic acid or DNA is a linear polymer of nucleotides, and two such polymers come together to form a double helical structure. Another important properties of DNA, which again we are going to exploit during its purification, is the heat denaturation. This is very similar to what we have seen in case of protein. So, in case of protein, if you heat them, the protein unfolds, and if you cool it, then the protein can again go back reversibly to form the folded protein. We see something very similar in case of DNA. So, the two strands of DNA are represented here. These two lines are the two strands of DNA which are held together by hydrogen bonds between the nitrogenous bases which are on the inside. If I heat a solution of DNA, what will happen is these hydrogen bonds will start breaking. So, the two strands will sort of peel away from each other. So, the more heat I give, the more hydrogen bonds will be broken. So, that at some point of time that all the hydrogen bonds will be broken, so that the two strands of DNA will be completely separated from each other. Now, if I cool this solution again, this is a reversible process. So, it can go back and form this double stranded DNA. So, the breaking of hydrogen bond and separation of the two strands is called denaturation, and formation of the hydrogen bond and formation of the double helical structure upon cooling down the solution is called annealing of the DNA molecule. So, it can be a reversible process, and this denaturation and annealing also depends on the length of the DNA and composition of the DNA, because in the previous slide we saw that GC pairs form 3 hydrogen bonds, whereas AT pairs form 2 hydrogen bonds. So, depending on how much GC content is there in the DNA, the amount of heat that will be required to denature a particular double stranded DNA will increase, and that can be easily measured by a very simple property called the melting temperature. So, what we are plotting here is, so suppose we have taken two different DNA solutions. Let us say the first one is A and the second one is B. When we heat them and we observe the absorbance of this DNA, so the absorbance will change, because the absorbance of it when it is double stranded and when it is completely isolated strands will be different. So, let us say the absorbance increases. So, as the DNA molecule denatures, more and more of the molecules are denaturing, the absorbance increases, and when it is completely denatured, all of it will be, so the will reach a saturation point. So, for two different DNA molecules or two different sequences, we see two different curves. The midpoint where we reach 50% denaturation. So, this is nothing but the middle point between this saturation point and this baseline. So, if we take as the 50% denaturation, the temperature at which it reaches this point is referred to as the melting point, and we see that the melting point is different for the two different DNA molecules. So, this melting point will depend on the length of the DNA molecule, and it will also depend on the sequence of the two DNA molecules. So, we are going to exploit this fact that DNA can denature and it can also be annealed back. What we are not going to do is we are not going to heat our solution, we are going to use chemicals to do the denaturation. Just like we saw urea and guanidinium hydrochloride can be used to denature proteins, we will see that the same chemicals or a different chemical can also be used to denature DNA molecules. So, in this week we are going to see extraction of DNA from cells, and we will mostly talk about extraction of DNA from bacteria. So, in case of bacteria, there are two different types of DNA. One is called the plasmid DNA and the other one is the genomic DNA. So, genomic DNA is the DNA which encodes all the proteins and other information for the normal function of the bacteria or the survival of the bacteria. So, it is its own DNA, and typically this DNA, if we talk about E. coli, will be something in the order of 5 times 10 to the power 6 base pair long. So, that is 5 million base pair long. Plasmid DNA are small circular DNA and they are much smaller. So, the typical size of a plasmid DNA will be in the range of 6000 base pair long. So, I am not going to talk much about plasmid DNA in this week's lecture, because we are going to cover we are going to talk in more details about plasmid DNA in next week's lecture when we talk about recombinant DNA technology. So, this is something that we can artificially introduce into a bacteria in order to make certain proteins. So, sometimes we might require to purify only the plasmid DNA, and some other times we may want to purify the genomic DNA. And another small information that, so this is a bacterial cell. So, it is a prokaryotic cell and this is a eukaryotic cell. What we want to see here is that if this is a cell, you see that in case of bacteria the DNA is here and all the proteins are here. So, everything is in the same place. So, when we break open this cell, all of them will be out in the solution, and from there we have to selectively purify the DNA. In case of eukaryotes, the DNA is in the nucleus, but again when we break open the cell membrane, we break open everything and all the cellular components are out in the solution, and from there we have to purify the DNA. So, it is quite a challenging task and we have to go through several steps to get a very high quality of pure DNA. So, what are the general steps? The first step, as we saw in case of protein purification, is that you have to isolate the cells. So, in case of protein purification, we were working with bacterial cells. So, in this case also we can use bacterial cells, but if it is some mammalian cell, then we will have to isolate the cells from the tissue from which the DNA has to be extracted. Once we have the cells, we will disrupt the cell membrane. In case of bacteria, we also have to break apart the cell wall, so that everything that was inside comes out into the solution. Now, this solution will have DNA, RNA, protein and all sorts of cellular debris. So, we have to selectively remove RNA, we have to selectively remove all proteins, and we have to also remove all the cell membrane components and other molecules. And finally, we will extract the DNA, the genomic or the plasmid DNA. Once we have our DNA, we have to make sure that it is pure. So, we will do gel electrophoresis to check the purity of the DNA, and we will also check the absorbance at 260 nanometers to check the concentration, which will tell us the amount of DNA that we have been able to purify. So, what is the source? The source of the DNA can be anything. So, it can be any living or dead tissue, and normally you have to extract out the cells from the tissue. So, these are all potential source for the DNA. And so here is one example where scientists have been able to successfully isolate DNA from fossilized animal remains, because as I mentioned that DNA is a very stable molecule, but depending on the quality of the DNA, it can be isolated. So, in case of laboratory, the common sources are different animal tissues such as liver, spleen, kidney, etc. And in case of bacteria, we take the whole bacteria. So, we can grow the bacterial cells in a culture, and depending on our requirement, we can purify the genetic or the plasmid DNA from the bacteria. In case of plants, we can also isolate DNA, but it poses some extra challenges. So, any part of a plant can be used, but the presence of cell wall, which is a very rigid structure which helps to maintain the shape of the cell in case of plants, and this high amount of polysaccharides poses extra problems. So, there are extra steps that you will have to go through to extract the DNA from plant sources. So, what are the steps? In this lecture I will just briefly go through the steps, and then I will talk about them in more details in the next lecture. So, if we take the example of bacteria, the first step will be to break down the cell membrane. This is something very similar that we did during protein purification. In case of protein purification, we sonicated the proteins, the bacterial cells, and we also used lysozyme. So, in this case, since we are going to use a very small amount of bacterial cells, we are not going to sonicate, we are only going to use lysozyme. So, lysozyme is an enzyme which breaks down the bacterial cell wall, and this is the enzyme which is actually found in our eyes also, and it actually protects our eyes from bacterial infection. So, we use the same lysozyme to break open the bacterial cells. Once everything is out in the solution, then what we need to do is we have to selectively separate DNA from proteins, because the DNA can be associated with different types of proteins. There are transcription factors, there are different proteins which bind DNA, and most of these interactions are charged interactions. Also, we have to denature the DNA. So, all of this is achieved by reducing or increasing the pH of the solution. So, what we do is we use a alkaline medium to break all the hydrogen bond interactions. We also increase the salt content so that these different ionic interactions are disrupted, and we also use EDTA, which is a small molecule that binds different ions such as magnesium ions, manganese ions. So, these are the ions which are removed so that different enzymes such as DNase, which can break a DNA, which can chew up our DNA. So, those enzymes are inactivated. Also, adding EDTA makes the cell wall of the bacteria unstable. So, it helps in both degrading the bacterial cell wall and also protecting the DNA from several enzymes. SDS is added to solubilize the membrane. So, this SDS is sodium dodecyl sulfate, and it is the same surfactant that we use when we run SDS page gel. So, we have seen there that it can denature proteins. So, when we add SDS, it disrupts the membrane and it also denatures the protein, so that all the protein DNA complexes that are present will be disrupted. High salt is used to again disrupt the protein DNA interaction, because these interactions are also driven by electrostatic forces. And finally, we add RNase to chew up all the RNA that is present in this whole cell lysate solution, because we saw in the previous slide that RNA is less stable, and RNase, this enzyme selectively degrades RNA molecules. So, the RNA will be chewed up, and that will help us to easily remove the RNA molecule. So, once we have all of this, once we have the cell lysate and we have successfully disrupted all these interactions, now what happens is that this cell debris, the denatured protein, the chewed up RNA, they will start to form aggregate, and to speed up the process, we put this cell lysate, the tube containing the cell lysate, into a centrifuge instrument, and we centrifuge it, so that everything is precipitated out, and the solution contains the DNA. Now, to purify the DNA and remove it from other small molecules and these monomeric nucleotides, we precipitate the DNA by some non-polar solvent. For example, we can use ethanol or we can use chloroform isoamyl solutions to precipitate the DNA and remove the supernatant, and now we have this white precipitate in these two steps, which will be mostly pure DNA. Or we can take this solution and pass it through a silica gel column, which selectively binds DNA, and all other soluble parts are washed out from the column. So, either using any of these two methods, we can purify the DNA selectively and get a very high quality of DNA. Once we have the precipitated DNA or we have the DNA bound to the silica column, we can resuspend it or we can elute it from the column using nucleus free water or some buffer. Typically a buffer is used which is Tris EDTA buffer at pH 8. So, now the DNA is in solution, we have to just do quality check and determine the amount that we have. So, the quality of the DNA is checked by running it on an agarose gel electrophoresis. Remember that you can run DNA in both agarose or polyacrylamide gel, but since these are very big DNA molecules, we are not going to use PAGE or polyacrylamide gel electrophoresis, we are going to use agarose gel electrophoresis. And that, if we see single band or very few bands that we can explain, we know that our DNA is highly pure, and then you can check the absorbance of your sample at 260 nanometer to determine the amount of DNA. What we also do is we also check the absorbance at 280, and then we take the ratio of these two absorbance. If the ratio is close to 1.8, it means that we are getting more absorbance at 280 at 260 than 280. So, it means that there is not much protein present in our DNA sample, and if the value is less than 2, then it also indicates that there is not much RNA contamination. So, gel electrophoresis will tell us the purity, and absorbance at 260 nanometer tells us the quantity, and also these two ratios, this ratio tells us that there is not much protein and RNA contamination in our DNA sample. So, again these are the references, and in the next lecture I will go through specific example of extraction of DNA, plasmid DNA from a bacterial source. Thank you.